Hello, Life Science. Hope you're doing well. Today, we're going to continue Chapter 6, Role and Components of the Immune System, Document 2, Blood Groups and Another Self-Marker. Please make sure you have your books opened in front of you as well as the notes. We have already seen in Activity 1 that the immune system distinguishes between self and non-self through markers located on nucleated cells, and these markers are the HLA markers. Are there other markers in the body that also help the immune system to recognize the non-self? Do red blood cells that are enucleated present self-markers? So let's see here. The enucleated red blood cells, of course, they present self-markers and they present lots of them. But there are two main important markers located on the red blood cells. The first type, antigens of the ABO system, and the second type, antigens of the hesus system. These two types of markers will be discussed now in this document. So let's start with the ABO system. This system is determined by a group of glycoprotein molecules called also antigens. The antigens are of three types, antigen A, antigen B, and antigen O, and they are present on the surface of the red blood cells or erythrocytes. So, these three antigens that are glycoproteins or protein with carbohydrate, a combination between protein and carbohydrate, they are coded actually by a gene that is polymorphic, and this gene has the three alleles A, B, and O. The allele A codes for the antigen or the glycoprotein A, the allele B codes for the glycoprotein B, and the allele O, if you remember, it doesn't code for any glycoprotein. So, the figure represents a red blood cell, and on the surface you see clearly the antigen that is also called agglutinogen. So the first human blood transfusions were often bound to fail until Karl Steiner demonstrated that some people's serum or plasma can agglutinate the blood of others. After that, he presented the blood grouping system, ABO system. So, what is agglutination? What do we mean when we say some people's plasma can agglutinate the blood of others? So, agglutination or hemagglutination, it is a complementary reaction between agglutinin and agglutinogen. The agglutinogens, they are located on the surface of red blood cells. And the agglutinins, these are proteins located in the plasma. And these agglutinogens and agglutinins, they are present in each individual since birth. So, you have the experiment presented in the table of document B. So please make sure you have your books opened on page 116 and look at document B. And I want you please to pause the video for a while and try to interpret the results shown in this document. So what did we do here? Actually, they mixed red blood cells of an individual with the serum of another individual. And these two individuals are sometimes of the same blood group or of different blood groups. So let's see. For example, in the first row, red blood cells, they belong to an individual of group A and the serum to an individual of group also A. And in the second row, it's between an individual of A and the serum of B. Also in the third row, the individual is of blood group B and the second one is of blood group A. And in the last row, the two individuals, they belong to the same blood group. In every case, the result was observed, whether there is agglutination or not. So how to interpret this document? Actually, when a serum of an individual of type A is mixed with the red blood cells of an individual of the same type A, no agglutination takes place. Similarly, there is no agglutination occurring if the red blood cells in the serum are taken from individuals of the same blood type B. So I took the first row and the last row. However, when the serum of an individual of type B is mixed with the red blood cells of another individual of type A, or vice versa, I'm talking here about second and third row, agglutination takes place. This indicates that agglutination occurs only if red blood cells of a given group, like A, are mixed with the serum of a different blood groups, like B. So this leads us to think that in our blood, the red blood cells, they carry antigens or agglutinogens that are different from the proteins or the agglutinins that are found in the serum. So the serum of every individual contains proteins called agglutinins or antibodies, as we said before. They are represented in the opposite figure. You see these antibodies. They are proteins having Y shape. And we're going to discuss in details the structure of these antibodies in document 6. The same blood does not contain an agglutinogen that is compatible with the agglutinin. Otherwise, agglutination reaction would take place. As you see in the figure below, the antibody has a shape that is compatible with the agglutinogen present on the surface of the red blood cells. So the result will be a reaction between the antibody and the agglutinogen, and this is what we call agglutination reaction. So if I have in my blood antibody and agglutinogen that are compatible, so agglutination would take place, and this, of course, leads to a blockage of the circulation. That's why the antibody and the agglutinogen should be different in the same blood. So this table summarizes very well the 4 blood types, A, B, AB, and O. So when erythrocytes or red blood cells carry on their surface antigen A, and the plasma surrounding these erythrocytes carries anti-B antibodies, the blood type will be type A. So the blood type A is characterized by the presence on the surface of red blood cells of type A antigens and anti-B antibodies. While when the erythrocytes carry on their surface antigen B and in the plasma anti-A antibodies, the blood type will be B. So type B is characterized by type B antigens on the surface and anti-A antibodies in the plasma. Now, if both antigens A and B are present on the surface of erythrocytes, and when the plasma has neither anti-A nor anti-B antibodies, the blood type will be type AB, and the erythrocytes so carry both antigens A and B, while the plasma has anti-A, anti-B antibodies. But in the last case, if the red blood cells carry neither antigen A nor antigen B, however, both anti-A and anti-B antibodies are present in the plasma, the blood group will be type O. So type O is characterized by the absence of antigens on the surface of red blood cells, but with the presence of both anti-A and anti-B antibodies. So, for successful blood transfusion, the antigen of the donor and the antibody of the recipient must be different. In other words, the antibodies present in the plasma of the recipient should not react with the antigens present on the surface of red blood cells of the donor. Otherwise, agglutination would take place, followed by hemolysis, that means destruction of the red blood cells. So, you have here an example between an individual who is a donor of blood group B and another individual who is a recipient of blood group A. By referring to the small table that you see on the left, the donor B, we care mainly about his antigens present on the surface of red blood cells. So the donor B, being of blood group B, what does he have as antigens? He possesses antigens B. Why? The recipient of blood group A, being of blood group A and being a recipient, so we care for what he has in his plasma. He possesses actually the antibody B. So the donor has antigen B, the recipient has antibody B, and since antigen B and antibody B are compatible and they can bind to each other, so agglutination would happen and transfusion of blood fails. Okay, why do we care for the antibodies of the recipient and for the antigens of the donor? Because mainly the blood of the donor is very rich in blood cells, in red blood cells, and when these red blood cells are transfused to the recipient's blood, so the blood of the recipient, rich in his plasma with antibodies, will receive these red blood cells. And when there is compatibility, there will be agglutination. So let's do an application here. In the first case, the donor O and the recipient A. So as we said, we care for the antigens of the donor and the antibodies of the recipient. The donor O has no antigens. The recipient A has antibody B. So since as you see no compatibility here between the antibody and the antigens, since no antigens, then transfusion can happen and it's successful. In the second case, the donor is B and the recipient is O. So in this case, the donor has antigen B and the recipient has both antibodies A and B. So necessarily, there will be agglutination between the antibody B found in the plasma of the recipient O and the antigen B found on red blood cells of the donor B. So in this case, the transfusion failed. Here is a scheme that shows the possible transfusions between the different blood groups. Blood group O is a universal donor since it has no antigens, and blood group AB is a universal recipient since it has no antibodies. So the figure shows clearly that every blood group can donate blood to the same blood group. So A can give to A, B can give to B, O to O, and AB to AB. You see that all the arrows, they originate from O. So O can give blood to A, to AB, and to B. However, you see that AB receives all the arrows. So AB can receive blood from A, from O, and from B. A can receive blood only from O, and B can receive blood only from O. Before any blood transfusion, it is necessary to determine the blood group of every individual. And this is what we call blood grouping. What does blood grouping consist of? It consists of using test sera containing anti-A or anti-B and to mix them with blood drops of an individual. According to the agglutination result, appearance of small granulated red clumps, the red blood cells agglutinogens can be determined. So, as you can see in the figure, if you see reddish homogeneous drop, so no agglutination, while if you see small granulated red clumps, then this is a sign of agglutination. Now, if you use the microscope, and in the case of agglutination, you see that the red blood cells are gathered and attached to each other, while in the case of no agglutination, the red blood cells are free away from each other. So, how to determine the blood group of someone? 3 drops of blood are obtained from the individual. We add to the first drop anti-A, anti-B to the second, and anti-A and anti-B to the third. The agglutination between the drop of blood, red blood cells antigens, and the antibody indicates the presence of the corresponding antigen. Example, agglutination between blood and anti-A indicates that this blood contains antigen A. If no agglutination occurs between the second drop and anti-B, then no antigen B in the blood, and that blood group would be A, like for the first individual in the figure. The third drop is used to make sure of the results of the first 2 drops. For the second individual, he shows agglutination with anti-B and with anti-A and B mixed together. Then he has antigen B and his blood group is B. The third individual, he shows agglutination with anti-A, then with anti-B, and of course with both anti-A and anti-B. So he has both antigens A and antigens B. Then he is of blood group AB. While an individual of blood group O, for example, will not show any agglutination with anti-A and with anti-B. Here is another figure that makes things clear to you. So the blood types AB, AB, and O. For example, if 2 drops of blood are taken from every individual of these 4 individuals and mixed with anti-A and anti-B, the individual of blood type A would show agglutination only with anti-A. The individual of blood group B would show agglutination only with anti-B. The individual of blood type AB would show agglutination with both anti-A and anti-B, while the individual of type O won't show any agglutination, neither with anti-A nor with anti-B. So now we're done with the first type of self-markers present on the surface of red blood cells, which is the antigen or agglutinogen. Let's move to another type of marker, the rhesus system. At least 30 commonly antigens have been found in human red blood cells. Most of them are weak and therefore of no importance. However, following the ABO system, the rhesus system is the second in importance in the immune responses. This system is governed by a gene having 2 alleles, rhesus positive and rhesus negative, with allele rhesus positive being dominant over rhesus negative. What does the allele rhesus positive code for? This allele codes for a glycoprotein, the agglutinogen or antigen rhesus, that is present on the surface of red blood cells of some individuals. Why do we consider it as an antigen? Since it is recognized by the immune system as a non-self. So as you can see here in the figure below, an individual who is rhesus positive shows agglutinogen D on its surface, while an individual who is rhesus negative shows no agglutinogens on the surface of red blood cells. To better understand the importance of the rhesus system, experiments are performed in document F on page 118. So please open your book to page 118 and, as usually, I'm going to ask you to stop the video for a while and try to do this interpretation by yourself on a paper. Okay. So, when the serum of a rabbit is mixed with red blood cells of a rhesus monkey, of rhesus positive individual, or of rhesus negative individual, no agglutination takes place. This indicates that the serum of the rabbit doesn't contain anti-rhesus antibodies. On the contrary, there is agglutination of red blood cells of the monkey and of red blood cells of a rhesus positive individual when they are mixed with the immune serum of the rabbit that has previously received the red blood cells from the rhesus monkey. And there is no agglutination of red blood cells of the rhesus negative individual. This shows that the rabbit produces and secretes anti-rhesus only after the contact with the rhesus antigen. Of course, it is given in the experiment that agglutination is the reaction between the antibody and its specific antigen. So we can know that when agglutination occurs, this means that an antibody is present in the serum. So this experiment shows that anti-rhesus antibodies are only produced when a rhesus-negative individual is in contact with the blood of a rhesus-positive individual. So, when an individual is rhesus-positive, he possesses rhesus antigen on his red blood cells, as you see in the table, plus, and he has no rhesus antibodies in the serum. In the second row, when the individual is rhesus-negative, he has no rhesus antigen on red blood cells. You can see the sign minus. And the rhesus antibody is also absent, but it is formed when the individual is exposed to the rhesus antigen obtained from someone that is rhesus positive. We should note here that a rhesus negative individual can donate blood to rhesus negative individuals and also to rhesus positive individuals. But rhesus positive individuals cannot give blood to rhesus negative individuals. To sum up, the presence of two types of antigens A and B on the surface of red blood cells is responsible for blood transfusion rejection. The antigen rhesus is also involved in rejection, but of second importance. These antigens are considered as self-markers. Of course, the antigen A, antigen B, and the rhesus antigen are all considered as self-markers present on the surface of red blood cells. What's the difference between the ABO and the rhesus system? They both have antigens, but the antibodies of the ABO system are present since birth, whereas the anti-rhesus antibodies are formed after a contact of a rhesus-negative individual with a rhesus antigen. Thank you, Life Science, for your attention. See you later.